Okay, so we go on with our next presentation. The next presenter will be Ram Kumar Shrestha from Nepal. He is a bachelor in agricultural science from Tribhuvan University in Nepal. As other members of the group, he also conducted an agricultural training in the Arava International Center, and currently a master student in the program here in Rehovot. Ram will present his work on the assessment of growth in planta of Acidovorax citrulli type 4, pilus and polar flagellum mutants by diverse pathogenicity assays. Please, Ram.
Thank you, Suki. Welcome to all of you in my presentation. Today I am going to present my research exercise. The title of my research exercise is Assessment of Growth in Planta of Acidovorax citrulli type 4 pilus and polar flagellum mutants by diverse pathogenicity assay. This work was conducted under the guidance of Dr. Burdman from Department of Microbiology and Plant Pathology. Firstly, I want to introduce the plant pathogen Acidovorax citrulli and the disease caused by Acidovorax citrulli. It is a gram-negative and biotrophic pathogen by bacterium. It causes seedling blight and bacterial fruit blots of cucurbit. As you can see, the symptom of seedling blight in cucurbit plants, cucurbit seedling, it starts from the water soak spot, especially in the cotyledon, which get enlarged and becomes necrotic. Then later the plant will die like this. You can see the symptom in fruit also in various cucurbit fruits. It causes the rotten cavity inside the fruit, causing fruit blots of cucurbits. During late 1980s, there was huge outbreak of this disease in various states of United States. After that, the disease took more importance, and it got more importance among the scientific community, and research started intensively after that. And we know that from previous report, it has huge yield loss potential, and it is also reported that in conductive environment of high humidity and with moderate temperature, the disease comes devastating with 100% yield loss potential. So this disease is a very devastating disease of cucurbits. In other hand, the management of this disease is limited with the cultural management schemes and use of some compounds like copper-based compound, which has less efficacy to control this disease. So this disease is threatening disease for cucurbit industry. In order to save the cucurbit industry from suffering from the loss caused by this disease, we have to develop new tools and techniques for management of this disease. In order to do so, we need to have detailed information and knowledge about the host pathogen and host pathogen interaction. When we look on the pathogen Acidovorax citrulli, and we see on the pathogen diversity at various parts of the world, we can divide this pathogen into two distinct groups according to its genetic similarities and differences and biochemical utilization feature. The group first is mainly associated with non-watermelon types of cucurbits, that is melon type of plants, while the group second is mainly associated with watermelon type of plant. And this pathogen depends upon the functional type 3 secretion system for pathogenicity and hypersensitivity response in host plants. That means when we alter the function, impair the functions of type 3 secretion system of this pathogen will not be pathogenic anymore for its host plant and will lose the ability to cause hypersensitivity response in its non-host plant. If we look on the morphology of this pathogen, it contains single polar flagellum with basal body, hook, and the filament Similarly, it also contains the type 4 pili. It is a unique proteinaceous hair-like appendages generally extending from the pole of the cell. And the type 4 pili has unique capacity to extend its length and retract its length. It provides unique capacity to the bacterium. The bacterium is motile by means of polar flagellum in liquid medium polar flagellum, and its helical motility, helical movement provides swimming motility for the bacterium, whereas pilus provide a unique flagella independent motility which is known as twitching motility. For twitching motility, it extends its pilus, length of pilus added on the surface and retract towards it, so it moves forward in the direction of the extending and retracting of the pilus. Beside motility, these pilus and flagellum are the multifunction appendages. It has multiple functions. These has multiple functions and acts as virulency determinants for many of pathogens. Literature suggests that in many of biotrophic and necrotrophic pathogenic bacteria, flagella are found to be required for invasion stage of the host. In contrast, when we see on the Acidovorax citrulli and some other necrotrophic bacteria like Dictea didanti and Pectobacterium caroteroborum, these bacteria needs flagella for its full virulence. That means if we impair the function of flagella, the pathogen reduce its virulence in host plant. If we talk about type 4 pili, this is also the multifunction appendage. Beside twitching motility, these appendages are involved in surface adherence to attach on the surface, colonization on it, biofilm formation, a special type of colonial growth of the bacterium on the surface, solid surface, genetic material uptake, that means horizontal transfer of gene and the transfer of resistance genes from one bacteria to another bacteria, and the virulence of bacteria. Particularly in Acidovorax citrulli, type 4 pilus are involved in surface adhesion and biofilm formation. Or mutagenesis-based studies conducted in our lab had demonstrated the role of type 4 pilus and polar flagellum for the virulence of this pathogen. That means these appendages are necessary for virulence of this pathogen in host plant. So our objective to conduct this study was to assess the growth in planta of and symptom induction ability of type 4 pili and polar flagellum mutants of Acidovorax citrulli in comparison with wild type strand. That means we wanted to compare the growth ability in planta of this mutant strand with the wild type pathogen, and also the symptom induction capacity of those. For this study, we took three mutants line, two from type 4 pili mutants and one for polar flagellum mutants with a wild type strand, that is M6. M6 was member from group host that is related to the melon or melon loving type of Acidovorax citrulli, which was isolated from melon fruit plant, melon fruit from Israel, and all the other mutant strands were generated on the background of this M6. The first type 4 pili mutant M6M, denoted as M6M, was generated on the background of M6, which was impaired in PIL-T functions, PIL-EM functions, and gene of PIL-EM functions. This gene encodes for type 4 pili assembly protein. So without the type 4 pili assembly protein, this M6M strand is unable to produce any type 4 pili in its surface. So it lacks type 4 pili at all. Similarly, M6T strand was impaired in gene PIL-T, which encoding a protein that carries nucleotide binding domain required for twitching motility. So it does produce the type 4 pili, but the type 4 pili is like a paralyzed and it cannot perform the twitching motility. Instead of it, the strand was also hyperpilated. That means the number of pili on the surface was more than the M6, that wild type parental type of strain. The flagellum mutant M6 flag was impaired in field C gene, which encodes for flagellin subunit of the filament of flagella. That means it produces the flagella without the filament. So it produces naked flagella and doesn't contain swimming motility due to lack of the filament unit. These bacterial strains were grown in nutrient agar. In case of mutant strain, we add kanamycin for selective media, and we grew them in 28 degrees centigrade for our experiments. As a plant, host plant, we choose melon and off-field cultivar of melon, which is found to be susceptible for this disease, and the plants were grown in greenhouse condition from 25 to 28 degrees centigrade. For seedling inoculation, we prepare seedlings of off-field cultivar of melon in greenhouse for 1 week in plain moss pit. For inoculation of this, we prepare 24-hour fresh culture of above mentioned strain, and the culture cultures were suspended in sterile water and adjusted to the 10 to the power 8 CFU per ml. Then 5 microliter of droplet of 10 to the power 8 CFU per ml of bacterial suspension was placed at the base of the seedling, stem of seedling, and then 25 gauge needle was passed through the middle of the droplet for inoculation. Then instantly we removed the droplet from the site of inoculation by towel paper. For seedling death assay, this experiment was designed to follow the disease development and symptom induction capacity of different mutant strains in comparison to wild type strain. In this experiment, sterile water was inoculated as negative control as inoculation of the bacterial strains, and seedling death was followed up to 12 to 14 days after inoculation. For bacterial growth follow-up in plant system, we designed 2 experiments. One is bottom inoculation experiment, and next one is top inoculation experiment. In bottom inoculation experiment, we did inoculation as described in previous section. Seedling inoculation was done as described in previous section, and we took 1 centimeter piece of stem from site of inoculation. Here we can see the site of, sorry. Here you can see the site of inoculation. We took the 1 cm piece from the site of inoculation, sterilized with 70% ethyl alcohol. Then the piece was homogenized in sterile water and serially diluted and plated in NA plate, that is nutrient agar plate, for bacterial colony count. Similarly, we took another piece of 1 centimeter piece of stem piece from 4 cm above the site of inoculation, and bacterial estimation was done as mentioned in above, and the concentration of bacteria was estimated at day 0, that is day of inoculation, 2, 4, and 6 days after inoculation. As I already mentioned, this is the second experiment to follow the bacterial growth in planta. In this experiment, we did all the techniques similar, but just the reverse the order of the inoculation site. We did inoculation on the top of just below the cotyledon leaves, and we followed the bacterial concentration from site of inoculation and 4 cm below the site of inoculation. We took the sample similar as before and plated them in nutrient agar plate. Here we took concentration of bacteria in day 0, day 2, day 4, and day 6 after inoculation. In foliage inoculation, we prepare plantlets, small plantlets having 3 true leaves by growing them in plain moss pit for 1 week. Then after the seedling were transplanted in a media containing 1:1:1 vermiculite and moss pit and sand in 1:1:1 ratio, and allowed to grow for the plant up to 3 true leaf stage. For inoculation of those mini plantlet, bacterial suspension of about 10 to the power 6 CFU per ml was prepared by 24-hour fresh culture of bacterial strain, and those suspensions were supplemented with 0.045% of seal weight in order to reduce the surface tension during inoculation. And melon plants, these 3 true leaves melon plant were dipped inside the suspension for 3 minutes. Then these melon plant were allowed to dry inside in the greenhouse condition for bacterial estimation, and then we estimated bacterial concentration from the melon plant. For bacterial estimation, we took 12 leaf discs of 7 mm diameter. As you can see from picture, we took leaf discs like this, from 4 from each true leaves, 4 leaf discs from each true leaves, and those discs were weighted, surface sterilized with 1% of sodium hypochlorite and plated as described above in our previous experiments. Here we have results from seedling death assay. We did inoculation with fairly uniform concentration of bacterium, and we have result from this. And in x-axis we plot the day after inoculation, and in y-axis the days of death of seedling, percentage death of seedling. As you can see, the death of seedling started earlier and reached maximum level and have much more seedling death in M6 strand, that is wild type of strand. In all remaining mutant strand, the death percentage of the seedling was lower than the M6 strand. This is result from one of our experiment, but we have similar result from multiple experiments, so we can rely on this data. And during experiment we saw that the symptom induction ability of among the mutant strand were different. M6M started symptom earlier, and the death, even you can see the death of the seedling were started earlier in M6M strand among the M60 and M6 flag. That means there is little bit difference between M6M, M6 flag and M60 during the symptom induction capacity. That means these strands are different. Here I have to mention that the M6M and M60 was impaired in function of type 4 pili. In addition to that, our research in our lab found that the M6M and M60 was also impaired in function, also altered in function of the polar flagellum, and M6M was able to swim faster than the M6 wild type of strand in vitro conditions. So it is quite reasonable to have higher seedling symptoms, higher seedling symptoms, and earlier seedling symptoms in M6M than other rest of two. Similarly, in M60, the flagellar functions was also altered, but in this M60 strand, the flagellar function was negatively altered. That means we didn't see. The report says that previous finding that says that the M60 was unable to swim in vitro condition, and the swimming motility was altered. So it is also reasonable to think that the M60 strand has less symptom induction capacity due to impairment in both flagella and the type 4 pili. So we can say that both of this appendage has functions during virulence in pathogen in host. These are few pictures from 6 days after inoculation. You can clearly see clearly see the difference between the wild type of strain and mutated strain with control. There is not such a difference between mutated strain, as you can see here in 6th day, but the wild type of strain is highly virulent in comparison to other. These are the results from bacterial growth follow-up from seedling inoculation, when we inoculate seedlings at the bottom of the seedlings, and this is the result from the site of inoculation, bacterial population from site of inoculation where we inoculate the seedling. At the day of inoculation, we don't have significant difference between the size of inoculation. At 3 days after inoculation, we find the significantly lower population of M6 flag. At day 6 and day 9, the M6 population was lower than the M6. M60 population was lower than the M6, sorry. And M population was fairly uniform, and there was not a difference between M6 and M6. When we look on the forcium above the site of inoculation, the difference between strain was more stronger at the site, forcium above the site of inoculation. Here we find at 3 days after inoculation, all the strain were lower than the M6 strain, that is wild type of strain. That means the mutated strain has lower growth ability within planta. In day 6, the strain M6 was able to cope the situation, and growth was similar as M6. M6F was similar as M6, but two mutants line M6M and M60 was lower than the M6 parental line. At day 9, the M60 was unable to do growth rate as done by the M6 strain. So here the more important thing is where the pattern of the growth of bacterium, not the when does it has lower population or higher. The pattern shows that, clearly shows that the mutant strain has lower growth rate in plant, so the population of this mutant strain were lower in the plant. When we look on the data from the inoculation at the top, we find similar result like reduction in growth rate of the bacterium. We didn't find any significant difference between the inoculum size and the population size at 2 days after inoculation, but there was significantly less population of M60 and M6 flag at 4 days after inoculation. 4 days after inoculation, similarly at 6 days after inoculation, M6 was really lower than M6, that is parental strain. Similarly, in forcium below the site of inoculation, we find this result at 2 days after inoculation. There was not significant difference between those strains, but 4 days after inoculation, M60 was lower than M6 strain, and at 6 days after inoculation, M6M and M6 flag was lower than the parental strain. So here we can clearly see that the growth rate of this mutant strain were lower than the parental strain in plant, and it is reasonable to think that the growth rate is lower and the seedling death percent is also lower, so there is correlation between death of seedling and the growth rate of a mutant strain in plant tissue. Despite of many effort in foliage inoculation, we failed to follow the growth rate of bacterium in plant tissue, and in addition to this, the type 4 pili mutants behave something differently, and we couldn't follow the development of this bacterial strain within plant tissue because the less consistency of type 4 pilus mutants. But from our experiment, we clearly see that out of 3 experiments, from 2 experiments, we find the variation in inoculum initial infiltrated inoculum size of the M6 flag and M6M within plant tissue. And here is the difference between when we inoculate with a fairly uniform population of the fairly uniform size of the inoculum, there was significant difference between the infiltrated population of the M6 flag and M6M. That shows there is some advantages of having flagella for M6 over the M6 flag to enter inside the plant tissue during our inoculation procedure. That means the M6 flag was actively inserting it inside the plant tissue and get protected against our surface sterilization by hiding it inside the leaf tissue. You can clearly see that the difference between we find the difference between M6 flag and M6 strain. The M6 strain, the day after two days after inoculation, the M6 strain inoculated plant looks really poor with pale yellow leaves, and M6 flag was still looking healthy because of this difference of initial inoculum. As a conclusion, what we can conclude from our experiment is flagellum and type 4 pili mutants have reduced virulence and in stem inoculation of melon seedlings. In seedling inoculation, this result was also proved by Ophir experiment of Ophir in seed inoculation techniques. So we can say that the flagellum and type 4 pilus mutants have reduced virulence in stem inoculation of melon seedling. From previous reports, we know that the growth ability of this mutant strain is not different from the growth ability of the wild-type parental strain in vitro condition. However, we find the different growth ability of this mutant strain in plant tissue. That's why we can say that even though the malfunctions of these appendages does not affect the multiplication of Acidovorax citrulli in minimal media and in vitro condition, it reduced the capacity of pathogen to proliferate in plant tissue, in planta. From our foliage inoculation, we can say that the flagellum of Acidovorax citrulli might has role during foliage inoculation procedure we followed. That means the flagella of M6 has advantages over the flagella mutant strain during our inoculation procedure to enter inside the plant tissue. In stem inoculation, the effect of loss of appendages are stronger on the bacterial population far from the site of inoculation. As you saw in our experiment, the difference between the bacterial strain in population was stronger from the at the far point from the site of inoculation when we move 4 cm up or 4 cm below the site of inoculation. The difference in growth rate of this pathogen is correlated with the seedling death, as we saw in our result. And late build-up of pathogen population in seedling are not effective to cause the death of seedling, which might be due to reduced vulnerability of seedling with increasing age. Here I have to mention you that with increasing age of seedlings, the seedling loses its vulnerability to this disease. So at advanced stage of seedling growth, the bacterial population developed inside the seedling will not have pronounced effect and will not cause pronounced disease symptom in the seedling. So we can say that the late build of population of this mutated strain within host plant is not able to cause the seedling death caused by original M6 parental strain. Thank you.
Thank you, Ram.
Thanks for the presentation. It was very nice to watch. I just have a question about the inoculum concentrations in your results.
Yeah.
That you did a comparison of the effect of the mutations on the bacteria,
yeah.
right?
Which one?
And it can be any one of these graphs, like, yeah. So why are the concentrations different? I mean, if you want to compare the mutations, couldn't you do it like in the same concentration? So
Nice question, but what we have to consider here is we firstly for equalizing the concentration at initial stage, we measured the OD, that is optical density of the bacterial suspension, and we plate the bacterial suspension in a plate. Then we after we count the bacterial colony from the bacterial suspension, for the bacterial suspension to measure the concentration. So we cannot find the accurate concentration, that is 1 into 10 to the power 8 for all of the bacterial concentration, or all of the strain at once, because it is like a estimation by looking at the OD of the solution.
so you measure it after.
So yeah,
the
I measured
concentration.
at the day of inoculation, and I count 2 days after inoculation. The actual concentration of the strain were counted 2 days after inoculation. So there is difference. So we cannot perfectly adjust the concentration.
And I have another question.
Okay.
That I see, I mean, I understand the benefits in agricultural applications, because if we have bacteria that have these mutations, so they're gonna, the growth is gonna, you're gonna have a, it's gonna postpone the growth, so you have a agricultural advantage to it. But how do you do this? Like, how do you do this mutation, the pathogenic bacteria?
It is not a way of thinking to cause to make the mutation on bacteria to have agricultural benefit. It's the way of studying the role of the type 4 pili. So if we know the type 4 pili and its role during the host pathogen interaction, and if we have some chemicals and some tools to like paralyze the type 4 pili in system, and to reduce the number of type 4 pili, and to dysfunction the type 4 pili, if we have chemical in future, we can apply it as a weak point of the bacterium. So we can hit on the target of weak point of bacterium. So we will have some technique to overcome the disease.
Well, your steam inoculation and foliage inoculation, steam inoculation work, but foliage inoculation does not work, yeah?
Foliage inoculation was not worthy.
Why didn't it work?
It is long to say that the foliage inoculation, why it did not work, because in foliage inoculation it is long procedure. It takes a lot of time, and we have data from some of foliage inoculation, but I suspect some faults within it, so I didn't present it here, because I started foliage inoculation after 11 a.m. from morning, and I inoculate the plant till 6 and 6:30 p.m. 6:30 at evening. So there was difference between the time of inoculation, and we inoculate bacterium in sequential order, first M6, M6M, like this. So we cannot assume that the stomatal open during all the hour of the day are not similar. So it might has advantages for one bacterium to enter inside the leaf tissue when it was inoculated at afternoon time, while the bacterial strain inoculated at late evening will have less advantage. So I cannot use this data anymore for the analysis here. So I didn't present the data.
Well, and one more question. And in seedling death assay, you waited to see the symptom for 14 days, yeah?
Yeah, I followed the...
How you decide within 12, what are the basis to say that it will die in 12 to 14 days? Does it happens in commercial scale that the plants, if infected with Acidovorax citrulli, it will die in 12 to 14 days?
Here we follow the sibling death up to 12 to 14 days, and we clearly see the dead seedling and thriving seedlings. But in commercial nurseries, the initial inoculation is really lower. As I mentioned, that the vulnerability of the seedling decreases with increasing seedling age. That means if the initial inoculation is really high, it builds up its population really lower rate, and the build-up of population will be later stage. So it is not efficient to develop such a devastating seedling death in commercial field. That is the weak point we have, because if we have initial inoculum in seedlings and we find the seedling death, it's okay, we can discard the seedling from the transplant house. But if it is latent inside the seedling and we don't observe any symptom, there is possibility of transferring these seedlings into the field, so it will causes devastating huge loss in field. So this is weak point we have here.
Okay, thank you.
Maybe I think you can do some kind of correction for the variation in initial inoculum concentration. If you see this data, M6 has some higher value of initial inoculum concentration. In your result also consistently M6 had a higher value in most of your slides. In shoot inoculum also, I think there was a variation. Maybe I'm not sure whether this small variation might have some significant effect on your result. In the shoot inoculum also there was a variation. Maybe you can do some correction for this initial variation. I'm not sure.
As I already mentioned, that it is very hard. It is almost impossible to find the exact inoculum size here, and here. So we have to rely on the estimate on the day of inoculation. We have to rely on the OD, optical density of the solution. But it is not only in. Sorry, I want to show a thing. It is not only the matter of inoculum size here. The main thing is how much bacteria are inside the plant tissue at the day of inoculation. Either it is becoming high and we find purely uniform, statistically uniform population here. It's okay, because during the puncturing of the stem there might be difference because of working with the hand. Sometime more duration of puncturing may have more advantage for this bacterium instead of this bacterium which has higher concentration at starting. So we mainly rely on this data, not in this data. So this data is showing that there is not significant difference between the inoculum concentration. This is just for showing. If we have inoculum concentration equal here, it doesn't make effects on it.
Maybe you can have, if you see this one at 0, yes, after inoculation you have some variation. For instance, the M6 has a higher standard.
Here we don't have a statistical difference, but what we can say here is it's not only from my experiment, it is also from previous experiment foliage done by Ofirvar. He compared this one mutants with this one with uniform concentration, statistically uniform concentration of inoculum at day 0, and he find the similar results. So we can rely on this, and it is from multiple experiments we saw the similar trend. so we can rely on it.
Okay, good. Just mine is a kind of suggestion
Yeah.
to maybe you can try a kind of correction at for the variation, like covariance or some kind of. Thank you.
Yeah, as Ram mentioned, I mean this. And he worked in this specific system, but and many other experiments with similar conditions were done in the lab. And I wish, I really, because I think Ram explained well the limitations. That I want to tell in many other people, including in the lab, but from other groups, even don't look at the concentration of the inoculum. They just rely on the OD measurements, and I think it's good that the Ram presents the data on the concentration of that he tested it, and he presented the data on the concentration of the inoculum. Now, when I see 7.2 to 10 to the 8 and 6.80 T to the 10 to the 8, I mean 80 10 to the 8, it's, I mean, everybody that works in phytobacteriology says that it's not difference on it. I mean, and also time zero, there is not a statistical significance, and you can see even from the time 0 point that the M6 flag, okay, at time 0, it's the purple one, it's higher than the green one. I mean, even that you can see, so, but it's not. I mean, there is no meaning to this because it's not significant. And these results are, I mean, repeated in all the experiments done by Ram. So I think it's, I mean, it's not they are not. If you will say that there is M6 has 7.2 to 10 to the 8, and one of the strains has 6.8 to the 10 to the 7, not to 10 to the 8, or even 2 to the 10 to the 8. I mean, I would take care of it. And in fact, this is one of the reasons why Ram is not showing the results of the foliage inoculation experiments, is that he didn't have, I mean, at the time 0, he see that there were some problems with this system. So he preferred not to, because then he found that it's something that was related or to the preparation of the inoculum or to the inoculation procedure, and not to the real difference in virulence between the strains, okay? So it's only just to stress this point. But I want to make just one question, because I did many questions to Ram, and we worked together, so I don't think, I mean, many of the questions that I already asked him, but just one question that I didn't is that when you look to this picture, and you can see, okay, in all the experiments that you show in seedling inoculation, in the top and in the bottom, and looking at the top and the bottom, you see some significant difference between some mutants and the wild type.
Yeah.
Okay, and there is a clear trend that the mutants are impaired in growth and movement, okay, in the seedling. But the difference, even significant, statistically significant, are not so dramatic in these experiments. Okay, you can see there's not so dramatic difference, but the difference in seedling death are really dramatic. If you remember the other graph, so it doesn't seem to me to be only a matter of bacterial counts in the tissue. Maybe there is something else there.
Yeah.
Do you think? What can be? I mean, not only a matter of numbers.
As I mentioned, that these appendages are multifunctional appendages, not only used for colonization, not only used for biofilm formation. It also used for the effective transport from the pathogen to the host, so it can manipulate the host system for the benefits of the pathogen. So it can cause the death of seedling. So the colonization itself is not making such a dramatic effect between the M6 strain and the rest of mutant strain. There might be the possibility of unable to transfer the effector molecules within host system. So it will not be able to manipulate the host system to cause it die and take it benefit before during multiplication. So this also causes the low rate of the multiplication might be also cause of the less ability to transport effector to the plant system, so it will not derive enough nutrient for growth in plant tissue. So there might be the reason, so we need to look on it for further research.
We have time for last question, please.
Here, as earlier Burdman mentioned, that even if there is significant, statistically, even if M6, the concentration of M6 and M6N, even if there is significantly, if there is a difference in concentration, it doesn't make difference, as far as I understand, as Burdman mentioned earlier. But in your data, even after 6 days and 9 days, there is no difference between M6M, M6T, and M6 flat. But there is difference between M6 and M6T.
Yes.
So how could you say that there is? You say that there is difference in graph, but not difference in concentration. Even if it is different in concentration, you say it doesn't make difference. But when you come to the graph, then you say there is difference. So this also might not be the difference. Then if you say there is no difference in concentration, then in your result, if there is different concentration, then this small difference also might not be significant then.
If I well understood your question here, what is make difference here is the trend, not particularly in which days which bacterium concentration is lower. If you look at this data, here is clear picture of and clear trend of lower growth of the mutant strain in comparison to the M6 strain. What we look here is the more in trend of the bacterial growth in the plant, not the at which days which bacterial concentration will be higher and which bacterial concentration will be lower. So we look for the trend of the bacterial concentration, and it is also from multiple experiments, so we can rely on it. Multiple experiments were showing the similar result. In other experiments, I did not present here, you can find the difference between the M6 and M6F in these days, not in these days. It doesn't make difference. Only looking at the trend of the bacterial growth.
Means the bacterial count doesn't make the difference?
But we look the trend, not the specific date, not in the specific date.
No, I just mean to say the concentration of bacteria, does it make difference or not?
Yeah, we are looking for it, so it's make difference, no? We are looking for it, the growth of type 4 pili mutants in plant.
No.
So we are looking for it, no?
If the concentration of bacteria makes difference, then if you said in concentration it doesn't make difference, but here it makes difference. So
what we look here is not here, but here. In the day of inoculation, this thing says us that in day of inoculation we have not significantly, statistically not significantly different population of this strain within plant tissue. It is outside the plant tissue. So it is more rely on our result, more rely on this, not in this. It is within plant tissue, we don't have significant difference between the bacterial concentration here. So it's well to interpret here.
I will take the chairman right to raise another question after the last one, maybe either to you or to Saul. Is there, actually it interacts with the first question that Evelyn asked before. Is there a way to utilize such mutants of the bacteria in order to interfere with the pathogenic effect of the wild type population of the bacteria?
For me? Want to ask me? I mean, I know the answer.
Okay, I will try to answer your question. What I want to say here is bacteria has dynamic nature. In environment, bacteria has dynamic nature. It can give up its resistance gene, whatever it wants to give another bacterium, and it can take from other. So in environment, bacteria are accumulating the gene that gives benefit to it, not the weakness. It gives,
Right.
it takes strength. So
Right. However, if you grow these bacteria, these mutants in lab and spread them in the field, will they compete with the natural population? Will they interfere the pathogenicity of the natural population?
In case of type 4 pili mutants, I don't think so. This will also, still this pathogen has pathogenicity. That means this pathogen is pathogenic. Still, it doesn't possess virulence, enough virulence as the wild type of strain do. But in case of type 3 secretion system, if we interfere the type 3 secretion system, the pathogen will dismiss its pathogenicity, will not be pathogenic anymore. So there is possibility of using the type 3 secretion mutants line as a biocontrol agent for the control of this bacterium. It is just a possibility. And one report from the China last year, if I remember well, from China last year, they reported that there is little bit efficacy of using type 3 secretion system mutants as a biocontrol agent for Acidovorax citrulli. Little bit. It is not well. So
no, the answer is right. I mean, if we spread these mutants, these specific mutants into the fields, we will go to jail because they are still pathogenic and okay, but potentially we can use or test the use of non-pathogenic mutants or even other Acidovorax avenae. I mean, there was also some work from one group in the US that they tested some Acidovorax related, some related bacteria from Acidovorax genus that are pathogenic on other plants, but not on watermelon and melon, and they work well to some level in some kinds of infections. But I mean, there is no way that they will get permission to spread these strains because they are pathogenic to other plants. Even many biological control agents, for example, a famous genus is Burkholderia. Okay, that there are some very efficient at least at greenhouse conditions, very effective strains of Burkholderia, but there is no way that they will get permission because there are some other Burkholderia, not necessarily these ones that are opportunistic pathogens of humans. So these are already enough to impede their utilization, even that these potential strains were not shown to be pathogenic on animals. So there, I mean, from there is the potential, but when you are using up even type 3 secretion mutants, they can say no, but in the field they can get from horizontal transfer type 3 secretion from other bacteria and then become pathogenic and maybe even strongly pathogenic. So it's a problem to use.
Okay. Thanks, Ram, and thanks to everybody.